Plastid. 67 languages. Plastid. Plant cells with visible chloroplasts. Scientific classification. Domain, bacteria. Phylum, cyanobacteria. Clade, plastid. A plastid is a membrane-bound organelle found in the cells of plants, algae, and some other eukaryotic organisms. Plastids are considered to be intracellular. Endosymbiotic cyanobacteria. 1. Examples of plastids include chloroplasts, used for photosynthesis, chromoplasts, used for synthesis and storage of pigments, leucoplasts, non-pigmented plastids, some of which can differentiate, and apicoplasts, non-photosynthetic plastids of apicomplexa derived from secondary endosymbiosis. A permanent primary endosymbiosis event occurred about 1.5 billion years ago in the Archaeplastida clade, land plants, red algae, green algae and glaucophytes. Probably with a cyanobiont, a symbiotic cyanobacteria related to the genus Gliomargarita. 2. 3. Another primary endosymbiosis event occurred later, between 140 to 90 million years ago, in the photosynthetic plastids Paulinella amoeboids of the cyanobacteria genera Prochlorococcus and Synechococcus, or the PS. Clade. 4. 5. Secondary and tertiary endosymbiosis events have also occurred in a wide variety of organisms, and some organisms developed the capacity to sequester ingested plastids, a process known as kleptoplasty. A. F. W. Schimper, 6, was the first to name, describe, and provide a clear definition of plastids, which possess a double-stranded DNA molecule that long has been thought of as circular in shape, like that of the circular chromosome of prokaryotic cells, but now, perhaps not, see a linear shape. Plastids are sites for manufacturing and storing pigments. Problems donating. Other ways to give frequently asked questions. We never sell your information. By submitting, you are agreeing to our donor privacy policy. The Wikimedia Foundation is a non-profit, tax-exempt organization. If you make a recurring donation, you will be debited by the Wikimedia Foundation until you notify us to stop. We'll send you an email, which will include a link to easy cancellation instructions. I already donated. How often would you like to donate? One time give monthly. Please select an amount, USD. The average donation in the United States is around $13. $2.75, $10, $15. $25, $50, $75. $100 other. Please select a payment method. Bank transfer, ACH. Maybe later. 1. Suzo RCN. 2. 3. Continue. Wikipedia still can't be sold. The 17th of October, an important update for readers in the United States. You deserve an explanation, so please don't skip this 1-minute read. We're sorry to interrupt, but it's Thursday, the 17th of October, and this message will be up for only a few hours. We ask you to react on the number of times you visited Wikipedia in the past year, and if you're able to give $2.75 to the Wikimedia Foundation. If everyone reading this gave just $2.75, we'd hit our goal in a few hours. In the age of AI, access to variable facts is crucial. Wikipedia is at the heart of online information, powering everything from your personal searches to emerging AI technologies. Your gift strengthens the knowledge of today and tomorrow. Just 2% of our readers donate, so if you have given in the past and Wikipedia still provides you with $2.75 worth of knowledge, kindly donate today. If you are undecided, remember that any contribution helps, whether it's $2.75 or $25. Proud host of Wikipedia and its sister sites. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Plastid types. Leucoplasts in plant cells. And other important chemical compounds used by the cells of autotrophic eukaryotes. Some contain biological pigments, such as used in photosynthesis or which determine a cell's color. Plastids in organisms that have lost their photosynthetic properties are highly useful for manufacturing molecules like the isoprenoids. 8. In land plants, edit. Chloroplasts, proplastids, and differentiation, edit. In land plants, the plastids that contain chlorophyll can perform photosynthesis, thereby creating internal chemical energy from external sunlight energy while capturing carbon from Earth's atmosphere and furnishing the atmosphere with life-giving oxygen. These are the chlorophyll plastids, and they are named chloroplasts, see top graphic. Other plastids can synthesize fatty acids and terpenes, which may be used to produce energy or as raw material to synthesize other molecules. For example, plastid epidermal cells manufacture the components of the tissue system known as plant cuticle, including its epicuticular wax, from palmitic acid, which itself is synthesized in the chloroplasts of the mesophyll tissue. Plastids function to store different components including starches, fats, and proteins. 9. All plastids are derived from proplastids, which are present in the meristematic regions of the plant. Proplastids and young chloroplasts typically divide by binary fission, but more mature chloroplasts also have this capacity. Plant proplastids, undifferentiated plastids, may differentiate into several forms, depending upon which function they perform in the cell, see top graphic. They may develop into any of the following variants. 10. Chloroplasts, typically green plastids that perform photosynthesis. Etioplasts, precursors of chloroplasts. Chromoplasts, colored plastids that synthesize and store pigments. Gerontoplasts, plastids that control the dismantling of the photosynthetic apparatus during plant senescence. Leucoplasts, colorless plastids that synthesize monoterpenes. Leucoplasts differentiate into even more specialized plastids, such as the aleuroplasts. Amyloplasts, storing starch and detecting gravity, for maintaining geotropism. Elioplasts, storing fats. Proteinoplasts, storing and modifying protein. Autanosomes, synthesizing and producing tannins and polyphenols. Depending on their morphology and target function, plastids have the ability to differentiate or redifferentiate between these and other forms. Plastoms and chloroplast DNA/RNA, plastid DNA and plastid nucleids, edit. Each plastid creates multiple copies of its own unique genome, or plastome, from plastid genome, which for a chlorophyll plastid, or chloroplast, is equivalent to a chloroplast genome, or a chloroplast DNA. 11, 12. The number of Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Genome copies produced per plastid is variable, ranging from 1,000 or more in rapidly dividing new cells, encompassing only a few plastids, down to 100 or less in mature cells, encompassing numerous plastids. A plastome typically contains a genome that encodes transfer ribonucleic acids, tRNA, S and ribosomal ribonucleic acids, RNAs. It also contains proteins involved in photosynthesis and plastid gene transcription and translation. But these proteins represent only a small fraction of the total protein setup necessary to build and maintain any particular type of plastid. Nuclear genes, in the cell nucleus of a plant, encode the vast majority of plastid proteins, and the expression of nuclear and plastid genes is co-regulated to coordinate the development and differentiation of plastids. Many plastids, particularly those responsible for photosynthesis, possess numerous internal membrane layers. Plastid DNA exists as protein DNA complexes associated as localized regions within the plastid's inner envelope membrane, and these complexes are called plastid nucleids. Unlike the nucleus of a eukaryotic cell, a plastid nucleid is not surrounded by a nuclear membrane. The region of each nucleid may contain more than 10 copies of the plastid DNA. Where the proplastid, undifferentiated plastid, contains a single nucleid region located near the center of the proplastid. The developing, or differentiating, plastid has many nucleids localized at the periphery of the plastid and bound to the inner envelope membrane. During the development/differentiation of proplastids to chloroplasts, and when plastids are differentiating from one type to another, nucleids change in morphology, size, and location within the organelle. The remodeling of plastid nucleids is believed to occur by modifications to the abundance of and the composition of nucleid proteins. In normal plant cells, long thin protuberances called stromules sometimes form, extending from the plastid body into the cell cytosol while interconnecting several plastids. Proteins and smaller molecules can move around and through the stromules. Comparatively, in the laboratory, most cultured cells, which are large compared to normal plant cells, produce very long and abundant stromules that extend to the cell periphery. In 2014, evidence was found of the possible loss of plastid genome in Rafflesia lagusa, a non-photosynthetic parasitic flowering plant, and in Polytomella, a genus of non-photosynthetic green algae. Extensive searches for plastid genes in both taxons yielded no results, but concluding that their plastomes are entirely missing is still disputed. 13. Some scientists argue that plastid genome loss is unlikely since even these non-photosynthetic plastids contain genes necessary to complete various biosynthetic pathways including hem biosynthesis. 14. Even with any loss of plastid genome in Rafflesiaceae, the plastids still occur there as shells without DNA content. 15. Which is reminiscent of hydrogenosomes in various organisms. In algae and protists, edit. Plastid types in algae and protists include chloroplasts, found in green algae, plants, and other organisms that derived their genomes from green algae. Muroplasts, also known as cyanoplasts or cyanelles, the plastids of glaucophyte algae are similar to plant chloroplasts, excepting they have a peptidoglycan cell wall that is similar to that of bacteria. Rhodoplasts, the red plastids found in red algae, which allows them to photosynthesize down to marine depths of 268 m. 10. The chloroplasts of plants differ from rhodoplasts in their ability to synthesize starch, which is stored in the form of granules within the plastids. In red algae, fluoridin starch is synthesized and stored outside the plastids in the cytosol. 16. Secondary and tertiary plastids, from endosymbiosis of green algae and red algae. A leucoplast, in algae, the term is used for all unpigmented plastids. Their function differs from the leucoplasts of plants. Apicoplast, the non-photosynthetic plastids of apicomplex are derived from secondary endosymbiosis. The plastid of photosynthetic Paulinella species is often referred to as the cyanella or chromatophore, and is used in photosynthesis. 17. 18. It had a much more recent endosymbiotic event, in the range of 140–90 million years ago, which is explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. The only other known primary endosymbiosis event of cyanobacteria. 19. 20. Etioplasts, amyloplasts and chromoplasts are plant-specific and do not occur in algae. Citation needed. Plastids in algae and hornworts may also differ from plant plastids in that they contain pyrenoids. Inheritance, edit. In reproducing, most plants inherit their plastids from only one parent. In general, angiosperms inherit plastids from the female gamete, where many gymnosperms inherit plastids from the male pollen. Algae also inherit plastids from just one parent. Thus the plastid DNA of the other parent is completely lost. In normal intraspecific crossings, resulting in normal hybrids of one species, the inheriting of plastid DNA appears to be strictly uniparental, i.e., from the female. In interspecific hybridizations, however, the inheriting is apparently more erratic. Although plastids are inherited mainly from the female in interspecific hybridizations, there are many reports of hybrids of flowering plants producing plastids from the male. Approximately 20% of angiosperms, including alfalfa, Medicago sativa, normally show biparental inheriting of plastids. 21. DNA damage and repair, edit. The plastid DNA of maize seedlings is subjected to increasing damage as the seedlings develop. 22. The DNA damage is due to oxidative environments created by photooxidative reactions and photosynthetic /respiratory electron transfer. Some DNA molecules are repaired, but DNA with unrepaired damage is apparently degraded to non-functional fragments. DNA repair proteins are encoded by the cell's nuclear genome and then translocated to plastids where they maintain genome stability / integrity by repairing the plastid's DNA. 23. For example, in chloroplasts of the moss Fisca patterns, a protein employed in DNA mismatch repair, MSH1, interacts with proteins employed in recombinational repair, Rec-A and Rec-G, to maintain plastid genome stability. 24. Origin, edit. Plastids are thought to be descended from endosymbiotic cyanobacteria. The primary endosymbiotic event of the Archaeplastida is hypothesized to have occurred around 1.5 billion years ago, 25, and enabled eukaryotes to carry out oxygenic photosynthesis. 26. 3 evolutionary lineages in the Archaeplastida have since emerged in which the plastids are named differently. chloroplasts in green algae and / or plants, rhodoplasts in red algae, and muroplasts in the glaucophytes. The plastids differ both in their pigmentation and in their ultrastructure. For example, chloroplasts in plants and green algae have lost all phycobilisms, the light harvesting complexes found in cyanobacteria, red algae and glaucophytes, but instead contain stroma and granithollacoids. The glaucocystophycean plastid, in contrast to chloroplasts and rhodoplasts, is still surrounded by the remains of the cyanobacterial cell wall. All these primary plastids are surrounded by two membranes. The plastid of photosynthetic Paulinella species is often referred to as the cyanella or chromatophore, and had a much more recent endosymbiotic event about 91–40 million years ago. It is the only known primary endosymbiosis event of cyanobacteria outside of the Archaeplastida. 17–18. The plastid belongs to the PS clade of the cyanobacteria genera Prochlorococcus and Synechococcus, which is a different sister clade to the plastids belonging to the Archaeplastida. 4–5. In contrast to primary plastids derived from primary endosymbiosis of a prokaryotic cyanobacteria, complex plastids originated by secondary endosymbiosis in which a eukaryotic organism engulfed another eukaryotic organism that contained a primary plastid. 27. When a eukaryote engulfs a red or a green alga and retains the algal plastid, that plastid is typically surrounded by more than two membranes. In some cases these plastids may be reduced in their metabolic and / or photosynthetic capacity. Algae with complex plastids derived by secondary endosymbiosis of a red alga include the heteroconts, haptophytes, cryptomonads, and most dinoflagellates, equals rhodoplasts. Those that endosymbiosed are green. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. An illustration of the stages of interconversion. In plastids, alga include the euglenids and chlorarachniophytes, equals chloroplasts. The apicomplexa, a phylum of obligate parasitic alveolates including the causative agents of malaria, Plasmodium spp, toxoplasmosis, Toxoplasma gondii, and many other human or animal diseases also harbor a complex plastid, although this organelle has been lost in some apicomplexans, such as Cryptosporidium parvuum, which causes cryptosporidiosis. The apicoplast is no longer capable of photosynthesis, but is an essential organelle, and a promising target for antiparasitic drug development. Some dinoflagellates and sea slugs, in particular of the genus Alizia, take up algae as food and keep the plastid of the digested alga to profit from the photosynthesis. After a while, the plastids are also digested. This process is known as kleptoplasty, from the Greek, kleptes, thief. Plastid development cycle, edit. In 1977 J. M. Whatley proposed a plastid development cycle which said that plastid development is not always unidirectional but is instead a complicated cyclic process. Proplastids are the precursor of the more differentiated forms of plastids, as shown in the diagram to the right. 28. See also, edit. Mitochondrion organelle in eukaryotic cells responsible for respiration. Cytoskeleton network of filamentous proteins that forms the internal framework of cells. Photosymbiosis type of symbiotic relationship. Notes, edit. A. Sometimes Ernst Haeckel is credited to coin the term plastide, but his plastide includes nucleated cells and anucleated cytodes. 7, and thus totally different concept from the plastide in modern literature. References, edit. 1 Sato N, 2007. Origin and Evolution of Plastids, Genomic View on the Unification and Diversity of Plastids. In Wise R. Huber J.K., eds. The Structure and Function of Plastids. Advances in Photosynthesis and Respiration. Volume 23. Springer. Netherlands. pp. 75102. Do. 10.101781402040610610_4. ISBN 978-1-4020-4060-3. 2 More Kr. Magnabosco C. Momper L. Gold Basark T. Fournier GP, 2019. An expanded ribosomal phylogeny of cyanobacteria supports a deep placement of plastids. Frontiers in Microbiology. 10.1612. Do. 10.3389/fmic.2019.01612. PMC 6640209. PMID 31354692. 3 Vries, Jan de, Gould, Sven B. 15 January 2018. The Monoplastidic Bottleneck in Algae and Plant Evolution. Journal of Cell Science. 131, 2 JCS 203414 do. 10.1042/JCS.203414. ISSN 0021-9533. PMID 28893840. 4 AB Merin, Berger, Noack, Eva cm, Glockner, Janot, Melkonian, Michael, 5 June 2007. The ancestor of the Paulinella. Chromatophore obtained a carboxysomal apirin by horizontal gene transfer from a Nitrococcus-like Proteobacterium. BMC. Evolutionary Biology. 7, 185. Bibcode, 2007 Massey, 7, 85M. Do. 10.1186/1148-7148-7-85. PMC 1904183. PMID 175500603. 5 AB Arcoda Alder, Jesus G. Esteban, Rocho, Diago, Mariel Hughes, Humad, Jean, 29 January 2014. The plastide ancestor originated among one of the major cyanobacterial lineages. Nature Communications. 5, 14937. Bibcode, 2014 Natco, 5.4937. Do. 10.1038/Ncoms 5937. ISSN 2041-1723. PMID 25222494. 6 Schimper, AFW. 1882, über die Gestalten der Starkebildner und Farbe botanisches Zentralblatt 12. 517578. 7 Haeckel, E. 1866, Morphologist Individuen erster Ordnung, Plastiden oder Plasma stuck in his General Morphology. Der Organismen BD1, pp. 269-289. 8-carat picozoans are algae after all. Study the Scientist magazine registered trademark symbol. 9-carat colatucudi, p. 1996, biosynthetic pathways of cutin and waxes, and their sensitivity to environmental stresses, pp. 83108. In Plant Cuticles. G. Castians, ed., Bias Scientific Publishers Limited, Oxford. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 10-carat AB Wise, Robert R. 2006. The Diversity of Plastid Form and Function. The Structure and Function of Plastids. Advances in Photosynthesis and Respiration. Volume 23. Springer. pp. 326. Do. 10.101781402040610610_1. ISBN 978-1-4020-4060-3. 11-carat wick, S. Schneeweiss, G.M., De Pamphilis, C.W., Muller, K.F., Quant. D. 2011. The Evolution of the Plastid Chromosome in Land Plants, Gene Content, Gene Order, Gene Function. Plant Molecular Biology. 76, 35273297 do. 10.1017/S11103-011-9762. 4. PMC 3104136. PMID 214248777. 12-carat wick, S. Norman, J. 2018. Molecular Evolution of Plastid Genomes in Parasitic Flowering Plants. Advances in Botanical Research. 85, 315347. Do. 10.1016/bs.erb.2017.11.014. ISBN 9780128134573. 13-carat AB plants without plastid genomes. The Scientist. Retrieved 26 September 2015. 14-carat Barbrook AC, Howe C.J., Pertan S., February 2006. Why are plastid genomes retained in non-photosynthetic organisms? Trends in Plant Science. 11, 21018 10.1016/j.plants.2005.12.004. PMID 16406-301. 15-carat DNA of giant corpse flower parasite surprises biologists. April 2021. 16-carat Viola R. Nival P. Peterson M. July 2001. The unique features of starch metabolism in red algae. Proceedings. Biological Sciences. 268, 1474 1417 22 do. 10.1098/RSPB.2001.1644. PMC 10885757. PMID 11429143. 17-carat Abilhi, Dukian, Ha, Chisan, Kim, Sunju, Park, Myungil, Bhattacharya, Debashish, Yoon, Hwansu, the 22nd of February 2019. Evolutionary Dynamics of the Chromatophora Genome in 3 Photosynthetic Paulinella Species, Scientific Reports. Scientific Reports. 9, 1 2560. Bibcode 2019 NAT SR 9.2560L. Do 10.101138/S41598-019-38621-8. PMC 6384880. PMID 307962445. 18-carat AB Gab, Arwa, Grossman, Arthur R. Bottasheria, Debashish, 5 May 2020. Palanik, B. ed. Paulinella, a model for understanding plastide primary endosymbiosis. Journal of Pharmacology. 56, 4. Wiley, 837843. Bibcode 2020 JPCGY 56837G. Do 10.1111/JPY.13003. ISSN 0022-3646. PMC 77348844. PMID 3228989. 19-carat Sanchez Baracaldo, Patricia, Raven, John Adot, Pisani, Davide, Noel, Andrew H. 12 September 2017. Early photosynthetic eukaryotes. Inhabited low salinity habitats. Proceedings of the National Academy of Sciences. 114, 37 E7737 E7745. Bibcode 2017 NAS 114 E7737S. Do 10.1073/NAS.1620089114. ISSN 0027-8424. PMC 5603991. PMID 28808007. 20-carat Luis Delay, Cecilio Valadez Cano, Bernardo Perez Zamorano, 15 March 2016. How really ancient is Paulinella? Chromatophora. PLOS currents. 8 do 10.1030/currents.tol.e68099364bb1a1299174b4e06b0c6b. ISSN 2157. 3999. PMC 4866557. PMID 28515968. Wikidata Q36374426. 21-carat Zhang Q, March 2010. Why does biparental plastide inheritance revive in angiosperms? Journal of Plant Research. 123, 2. 2016. Bibcode 2010 JPLR 123201Z. Do 10.1075-009-0291Z. PMID 20052516. S2 SID 51082244. 22-carat Kumara, Oldenburg DJ, Bendikirch, December 2014. Changes in DNA damage, molecular integrity, and copy number 4. Plastide DNA and mitochondrial DNA during maize development. Journal of Experimental Botany. 65, 22 6425 39. Do 10.1093/JXB/ERU359. PMC 4246179. PMID 25261192. 23-carat Oldenburg DJ, Bendikirch, 2015. DNA maintenance in plastids and mitochondria of plants. Frontiers in Plant Science. 6. 883 do. 10.3389/FPLS.2015.00883. PMC 4624840. PMID 265799143. 24 Udahara M. Kishita Y. Sekine Y. August 2017. MSH1 maintains organelle genome stability and genetically interacts with RECA and REC in the Mosfiska mitrella patterns. The Plant Journal. 91, 345546 do. 10.1111/TPJ.13573. PMID 2840783. 25 Arcoda Alder J. Esteban R. Diago ML. Humard J. September 2014. The plastide ancestor originated among one of the major cyanobacterial lineages. Nature Communications. 5, 4937. Bibcode, 2014 Natco, 5.4937. Do, 10.1038/MCOMS 5937. PMID 252222494. 26 Hedges SB, Blair J.E., Venturi ML, Shu J.L., January 2004. A molecular timescale of eukaryote evolution and the rise of complex multicellular life. BMC Evolutionary Biology. 4, 2. Do. 10.1486/1471-2148-4-2. PMC 341452. PMID 15005799. 27 Chan CX, Botticelli D. 2010. The Origin of Plastids. Nature Education. 3, 984. 28 Watley, Jean M. 1978. A Suggested Cycle of Plastide Developmental Interrelationships. The New Phytologist. 80, 3489. 502 do. 10.1011/J.100437.1978.TB01581.X. ISSN 0028-646X. JSTA 2431207. Further reading, edit. Hansen Mr., Kohler R.H. A Novel View of Chloroplast Structure. Plant Physiology Online. Archived from the original on 2005-06. 14. Wycliffe P., Sitban F., Wernerson J., Ezkara I., Ellerstrom M., Rusk L., October 2005. Continuous expression in tobacco leaves of a Brassica napus penthomolog blocks differentiation of plastids and development of palisade cells. The Plant Journal. 44, 1. 115. Do. 10.1145/J.1003X.2005.02482.X. PMID 16167891. Berkey C.W., 2001. The Inheritance of Genes in Mitochondria and Chloroplasts, Laws, Mechanisms, and Models, PDF. Annual Review of Genetics. 35, 12548. Do 10.10.146/anav.janae.35.102401.0902331. PMID 11700280. Archived from the original. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. PDF, on the 22nd of June, 2010. Retrieved the 1st of March, 2009. Chan CX, Bhattacharya D., 2010. The Origins of Plastids. Nature Education. 3, 984. Bhattacharya D., ed. 1997. Origins of Algae and Their Plastids. New York, Springerverlag/Wayne. ISBN 978-3-211-83036-9. Gould S.B., Waller R.F., McFadden G.I., 2008. Plastid Evolution. Annual Review of Plant Biology. 59, 491517. Do 10.1000.146/anav.rplant.59.0326007.0929115. PMID 183155522. S2 SID 304581113. Keeling P.J., March, 2010. The Endosymbiotic Origin, Diversification and Fate of Plastids. Philosophical Transactions of the Royal Society of London. Series B, Biological Sciences. 365, 1541 72948 do. 10.1098/rstb.2009.0103. PMC 2817223. PMID 201243441. External links, edit. Transplastemic plants for biocontainment, biological confinement of transgenes, co-extra research project on coexistence and traceability of GM and non-gram supply chains. Tree of Life Eukaryotes. Categories: organelles, plant physiology, photosynthesis, endosymbiotic events. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF.